teaching, forgetting to start recording. We got like another minute or so. I wanted to give a couple of announcements though. I'll use this time for that. You guys have an exam on Wednesday, and that will be our fourth exam. Then you have your final exam, and that is next week. I believe that is on I'm looking at it on my Canvas site. Let's see, that's the, I think that it's the 9th. Monday, Tuesday. Um, Monday. Okay, yeah, Monday tonight. Thank you, Morgan. Um, Zoe, yes, I have to adjust the times. The fourth exam is going to be open until 11 p.m. So you'll have all day to complete that. Um, I wrote your exam. I've written your fourth exam and I've written your final exam. I posted an announcement yesterday with some what I think is valuable info in terms of like preparing for your final exam. Um, if you're at all curious about grading, um, and what grades can replace what, um, I'm glad to talk to you on uh, I'm glad to talk to you in a Zoom meeting. Um, I'm going to try to put together a video. Okay, I'm going to make a very sincere effort to put together a video today explaining the grading. Um, I know it's in the syllabus, but I understand also that some folks might be like, yeah, but I just want I want something more tangible. I'm going to try to do that today. I'm going to make a very concerted effort to do that. Um, I apologize for not being able to definitively say, yes, I'm going to do this. But yeah, I'm just going to try to do that today. Um, in the event that I don't, I apologize. Just contact me, and it's actually a little bit easier for me to carve out time for a Zoom meeting. I understand, yeah, it's the same thing, but I'm going to try to record that video explaining the grading today. So then you can kind of assess, like, okay, you know, do I even, do I want to take the fourth exam? Is it worth my effort, or is it, should I just go ahead and reassess and reevaluate and just take the final? Um, this is kind of a general blanket statement. Um, your final exam is worth, I think, 15% of your grade. So if you do not take the final exam, you will get a zero for it. Um, so another way to say that is the final exam is mandatory. I don't have any policy like if you don't take the final, you get an F in the class or anything like that. Um, but if you don't take the final, you will get a zero for that portion of your grade. Now, the flip side of that is your final exam, if it's higher than all of your other in-class exams, um, the final exam grade will replace all those. Like I said, I'm going to try to make a video describing all that and kind of providing like itemized, like basically receipts of here's, if you do this, then this, and if you do this, then this. So at any rate, that's what I'm going to try to do today. Um, there is a practice exam for exam number 4 that is up. I would recommend doing that. You can see the sorts of questions, see some of the content. Um, and other than that, that's what I've got to say as sort of announcements. Um, today's meeting is going to be kind of a last day hodgepodge, hence the title, Last Day Hodgepodge. I'm going to post this as a PowerPoint later today as well, just because this is kind of like collecting from a couple of different places. So with all that said, I hope that you enjoyed your break. We're back for, like 4 days of actual classes and then finals. So I understand this is a stressful time, and so I hope that you're, taking care of yourself and giving yourself some time. Do we get a cheat sheet for the final exam? You can use a 3x5 index card, just like we have all semester long. Um, uh, so yeah, you can use that. Cool. Awesome. Oh, and sorry, just as a little bit of an aside, um, Congratulations to the women's volleyball team, the Texas State women's volleyball team. They're in the big tournament. I apologize for not knowing the better language, but congratulations. Good luck. Okay, so last day hodgepodge. We're going to get through membrane structure and assembly. What are the components of a membrane? First of all, we know that it's a lipid bilayer. And so what that phospholipid bilayer is commonly known as is a fluid matrix. Now, the lipid bilayer is a two-dimensional solvent. Okay, so what that means is that there's lots of stuff that's basically inserted into it that are going to be embedded in a membrane and they're going to show up on different sides of a membrane. Now, if we look at this model right here, what I want you to recognize is that there are things that are kind of on the outside, that outer leaflet, and then there are things that are on the inside. And I think that it's valuable to recognize that lipid bilayers and membranes are asymmetric. So we have a bunch of different components in this, and I want to highlight a couple of those. So first of all, I want to highlight things that are exclusively on the outside of a cell. We've got our oligosaccharide chains that are affixed to glycoproteins. Then we also have glycolipids. Okay. So if you look at, or if you think um, outer leaflet, I want you to think sugars. Now, these sugars that are on the outer leaflet, they are synonymous with communication. There are major parts of receptor proteins, receptors of like insulin and caffeine. And what they do is they're going to communicate with something or stimulate and react with something that's outside of the cell to communicate something to the interior side of the cell. Now, on the interior side of the cell, what we have is we've got sterols. Well, actually, we've got sterols on both sides, in the inner and outer leaflet. So these would be our cholesterol molecules. Now, these things are going to make your membrane a little bit more or a little bit less uh, flexible. Now, a higher concentration of sterols and cholesterol is going to make those membranes a little bit more rigid. Now, we also have a couple of different proteins. And notice this, 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 and this. These are all proteins. And what I want you to recognize about these is that they all have kind of different levels of integration into a membrane. If we look over on the far right-hand side, I'm going to indicate this with a star. This is an integral protein. So this is a protein that's integrated into the membrane, into the lipid bilayer, and it has what are known as multiple transmembrane domains or multiple transmembrane helices. So think about this as a single polypeptide. Here's the N termini, here's the C termini. Now that is one way in which an integral protein with multiple transmembrane domains can be kind of produced. Then I want to draw your attention over here, where I'm going to put a circle. This is an integral protein with a single transmembrane helix. Now, this single transmembrane helix is going to have, well, an N termini and a C termini, or an N termini and the C termini. So these orientations are going to be, they can be different from one another. So the single transmembrane domain, the N could be on the outside or on the inside, and vice versa. There are also peripheral proteins right here and here. So that word peripheral means they're kind of like tangentially connected. And one way in which they can be linked is through a covalent modification to a lipid. So essentially, a protein gets lipidated. A fatty acid gets covalently bound to that protein, or a glycolipid gets covalently bound to that protein, and it finds its way into, or a major part of the interior side of that lipid bilayer. Over here, where I'm going to use a square, we have a protein that's a peripheral protein that basically has a more transient or more temporary interaction with the lipid bilayer. So its contacts right here and right here and right here, maybe they're a little bit more dynamic. Maybe they're a little bit less stable in the sense that they're there for a little bit, but then not for long. Now, what's significant about these proteins is that these peripheral proteins, think about this as like a reversible modification. They can be cut or whatever links them to the lipid bilayer here, here, here. Those can all be released, in which case that protein is no longer associated with a membrane. Now, add to the chat if anyone has ever heard of the aging disorder, progeria. Has anyone ever heard of progeria? I'm going to take a sip of my coffee while people answer.
I just know I was trying to track whatever I was asking and there were some.
Okay, so progeria is an aging disorder that someone when they're like 8 years old, they might appear to be like 60. Um, and whenever I was in middle school, there was a girl who had progeria, and I think she ultimately passed away when she was about 13 years old. But she was experiencing things that most geriatric folks experience when she was like 10. So it's it, progeria is a disorder or disability that is associated with a protein that gets associated with a membrane. And at some point, it should release or it should be released in order to then go on into the cell and modify transcription or translation or do something within the cell. But if it doesn't get released, then it can't carry out its function. Okay, so think on the outside of the cell or the outer leaflet, think sugars, think glycoproteins or glycolipids. That GLYCO is your sugar indicator, glyc. I think that's Greek. Then on the interior side of the cell, well, we have our integral proteins, so it's integrated within the lipid bilayer. And then we have our peripheral proteins that either have a covalent modification or some other sort of more reversible or transient interaction with the lipid bilayer on the interior. Okay, now this is just another depiction of those same sort of things that we're kind of seeing here. Integral proteins are associated with tightly or are associated very tightly and requires a detergent from like a um, from the standpoint of like what am I thinking um, a purification or an analysis standpoint. What you need to do, since they're a part of a membrane, well, membrane is a super hydrophobic thing. So if you want to release or study that protein, you got to really bust it up with something like a detergent. Okay. Now, when we have these proteins that are integral proteins, they're found in a couple of different classes. The N termini can be on the inside or the outside. Here we see that. A type 1 integral protein has the N termini on the outside and the C termini on the inside. A type 2 has the N termini on the inside and the C termini on the outside. Type 3 is a single polypeptide, the N and C termini on opposite sides. A type 4 is um, multiple polypeptides that assemble. Now, this isn't, I don't want you to commit these to memory, but instead what I want you to do is I want your attention to be drawn over here. This region, which I'm going to highlight with the letter A, within our lipid bilayer here, add to the chat, would this be a polar or a non-polar region? Absolutely. It's a very non-polar region. Okay. So when we look at all of these different proteins, we have a way to kind of, if we look at the primary structure of a protein, we have a mechanism that we can use to predict whether or not a transmembrane domain is present. So if we look at the primary structure, so the amino acid sequence, we can look at the amino acid sequence, and if we see a dense packaging of non-polar a dense packaging, a cluster, I guess you could say. If you see like 15 or let's say 20 amino acids in a row and they are non-polar, you have pretty good reason to believe that that's going to be a transmembrane domain. Now, this is something that we actually looked at previously. Does anyone remember the blank plot that looks at the primary structure of a polypeptide to tell you and classify if something is polar or non-polar? Hydropathy plot. Absolutely right. Great job, Micaiah. So, a hydropathy plot is a tool that you can use to analyze the primary structure of a protein and say, "Oh, okay, well, we've got 15 amino acids in a row that are all nonpolar. Okay, I think that those are going to end up as a part of a transmembrane domain. And if we look at this specific sequence, phenylalanine, glycine, valine, methionine, methionine's not, that's decently polar. It's got the sulfur, but still there's a methyl group. Alanine, glycine, valine, isoleucine, glycine, threonine. Okay, so threonine is a polar residue, but it's a polar uncharged residue. So there are certainly some outliers. For instance, over here, we've got a glutamate, histidine, a serine. These are polar residues and some even polar positive residues, but by and large, the amino acids that make up a transmembrane domain are largely going to be nonpolar. And that's a useful tool. Okay, so in addition to helices, and thinking about secondary structure of proteins, a transmembrane domain is not just going to be an alpha helix or alpha helices. Instead, you can have beta sheets that kind of coil up to make what's known as a beta barrel. Because this, if you look at this maltoporin, for instance, that's a bunch of beta strands. And those beta strands, they kind of coil around one another. And if you were to look at any one of these proteins from the top down, which is what you see right here, you would see there's a central cavity and there's a pore there. And that pore, well, chances are that pore is a part of a channel, in which case ions can make it through there. So transmembrane proteins are not just alpha helices. Transmembrane proteins can be alpha helices or beta sheets. Now, so we've talked about integral transmembrane proteins, but now let's look at um, our peripheral proteins. So some proteins are linked to fatty acids or lipids in general. They're covalently bound to lipids that anchor to a protein. For example, this right here, this molecule right here, that's a long hydrocarbon chain. Geranyl is also a long hydrocarbon chain. And these chains right here can be added to a protein and basically anchor that protein in a membrane. Now, this would be a farnesyl residue. This is a geranyl residue. And then this right here, this whole thing is what's known as a GPI. And I'm going to come back to that in just a moment. Now, there are a number of different linkages, lipid linkages, four major types of lipid linkages that have a protein that associates with um, has a protein associate with a membrane. We have that geranyl or farnesyl residues. These molecules on the left, they can be added to a cysteine. So basically, here's your protein, and then there's a carboxy terminal cysteine, and that cysteine, rather than being SH, rather than it having that, instead, what we get is we add this thing to that S. So you get something that looks like this. Yeah, that's not drawn well. I apologize. But you get something that looks like that. And that molecule can be integrated into a membrane because it's super hydrophobic, in which case that protein right here that I've circled is basically kind of anchored, it's nailed down to that membrane. Now, there are other instances where fatty acids, like here's palmitic acid or myristic acid. Now, I never asked you to know specific fatty acids, but those are, I think, 16 and 18 carbon long fatty acids, which get added to a glycine or a cysteine or a serine, and that hydrophobic molecule gets anchored in that membrane. Then, last but not least, is this one where here's our protein. Okay. Our protein has a modification to it. And that modification, so we'll jog back. So here's that same protein that I just circled. We have a phosphoethanolamine. And that phosphoethanolamine gets a series of sugars added to it. And then last but not least, you get this phosphatidylinositol. So this would be like a protein. Think about it simply as a protein plus a um, glycerophospholipid. Okay. So that protein plus that glycerophospholipid, well, these tails right here get anchored and inserted to the membrane. Now, what I want you to take away from all of this is that the these are examples of post-translational modifications. So lipids and fatty acids can both be added to an amino acid, and that protein then gets associated with the membrane. This would be an example of largely those peripheral proteins. Okay. Now, this is just some examples of thioester link of what these bonds actually are, and just some examples and details of those. Now, I don't want you to uh, stress about GPI, but I do want you to think about these amino acids and what make them kind of unique from one another. Cysteine and serine, have we talked about modifications of cysteine of serine? Yeah, phosphorylation. Serine is a great target for phosphorylation. Well, serine can also get a um, fatty acid or lipidation is what this is called. Lipidation. Okay. Now, does anyone have any questions about membrane proteins? If any questions about membrane proteins come up, just feel free to go ahead and drop that into the chat. Whether it's a private message or a group message, it's fine with me. I'll do my best to answer your question. But now moving on to the next component, transport. So transport of molecules across a membrane is a pretty important subject. Now, transport of molecules may either be mediated or non-mediated. There are some molecules that can simply diffuse through a lipid bilayer. Now, that would be an example of non-mediated transport. Now, the rate of diffusion depends on the concentration gradient and the substance's solubility in the membrane's nonpolar core. So if something think about it as, oh yeah, it can maybe move across, but then once it hits this non-polar core of your lipid bilayer, maybe it runs into some problems. Maybe it runs into some challenges. Like it's a superpolar molecule in a non-polar region, and all of a sudden it's like, I can't handle this. I can't handle this. And it basically stops off there. So you've got to have something that's both well soluble in a polar and a non-polar environment, or at least semi-soluble. Now, water, oxygen gas, and steroids readily diffuse across a membrane. There is also what's known as mediated transport. Now, there are two major categories depending on the thermodynamics of that system in question. We have passive mediated transport or facilitated diffusion right here, where molecules flow from high concentrations to low concentrations. And in some cases, or that in facilitated diffusion, we've got a protein that's kind of helping out with that. Now, there's a last mechanism for transport, and that is what's known as active transport. In an active transport, you're using something like the hydrolysis of ATP to move something against its concentration gradient. So these green boxes, they're going from low concentration, low, to high concentration. Moving from that low to the high concentration, well, it's not the direction that they want to go. And so in order for that to happen, you've got to use something like ATP to provide the energy to pull it across. Now, whenever we talk about that transport, well, proteins are typically going to facilitate that, and proteins are going to increase the rate of that transport. Now, solutes can either go along their concentration gradient, in the case of facilitated diffusion, or against their concentration gradient, which is what we see in active transport. Now, typically there are three major proteins that you need to, and I would say proteins and I guess kind of styles or methods of transport. You have something like a uniport, where a single molecule is moving in one direction. Okay. There's also a symport, where you are moving two molecules in the same direction. Hence, same. Then last but not least, you have an antiport. Sometimes you'll hear these referred to as antiporters, and these are proteins that are moving two things in opposite directions, hence the anti. So these two things moving in opposite directions, well, there might be a kind of reason for that. In addition, when it comes to transporting different molecules across membranes, this can get really pretty complicated and hairy because you've got lots of different things going on with, oh, well, this is going in this direction, that's going in that direction, and that's a part of a greater system. Now, there are also proteins that you might know as carriers or channels. Channel transporters have a pore through which the solute passes at a very high rate. Think about those beta barrel proteins that we saw in the previous few slides. Well, they have a hole that basically opens up and things can flow through. Now, these are typically very selective and they tend to be gated. What that means is that there's another part. So I'm going to run back real quick. When we look at something like this, you might have one of these groups right here, one of these, random coils on top of the protein. What it might do is it might kind of act as a lid. And in the presence of either a specific voltage, it might be closed off. And then in other instances, it might open up. So that's one way in which gated channels function. Upon stimulation, they'll open up so that something can move across and move through. They're going to be very selective. One example of these are porins. I want to talk about porins in just a moment, but carrier transporters, by contrast, bind solutes on one side and undergo a structural change, releasing the solute on the other side. And a great example of these are your GLUT transporters or your glucose transporters. These are sometimes known as translocases and permeases. Now, there are, when it comes to these transporters, one thing that's important to understand is that this is the way that lots of drugs are designed. For example, transport inhibitors. There are SSRIs, or does it add to the chat if anyone knows what an SSRI is? Um, so essentially, this is the mechanism by which oh, sorry, yes. Thank you, Morgan. An SSRI is a selective serotonin reuptake inhibitor. I understand that. Yes, you answered my question. Sorry about that. Not a good question. But thank you. Yes, absolutely right. So selective serotonin reuptake inhibitor. And essentially what we have is serotonin, which is synthesized from tryptophan, one of our proteinogenic amino acids. Well, it gets packaged up into these vesicles. These vesicles will merge with the membrane or the plasma membrane of a neuron, and that serotonin will be released into the synapse where another adjacent neuron will receive that serotonin. And then after a little while, that serotonin will be released and it will go back into the initial neuron. Now, drugs, or an example of a drug that's a serotonin reuptake inhibitor, what it's going to do is it's going to block this step right here. It's not going to produce more serotonin, but instead it's just going to block that off so the serotonin doesn't get packaged back up. Instead, it kind of sustains a stimuli. Now, um, so that's one thing that these drugs can do. Um, Now, the next group of proteins that I wanted to talk about are porins. Okay, so this right here, I remember the first time I saw this structure, I was like, okay, cool. I see there's a pore, there's a pore, there's a pore. And that was an incorrect evaluation of this. So the pore that's in question is this guy right here. Now, this is a pore that's made from basically three beta barrel proteins. And essentially, these proteins, what they'll do is they'll undergo conformational change. I've drawn these arrows in to indicate that they rotate. And when they rotate, what happens to that central pore, or that central cavity, it opens up a little bit, enabling something to move through in the space between the membrane or um, the beta barrels. Now, this is basically the simplest membrane transporter. They're located in the outer membrane of bacteria, mitochondria, and chloroplasts. The structure of a porin determines its selectivity. Now, these are typically going to be considered always open in general, but sometimes they can kind of close off by doing these rotations. The porin that I want to talk about specifically is a porin known as aquaporin. Aquaporin is just a fascinating protein because to me, it takes me back, and I won't elaborate too much, but this takes me back to like Gen Chem 1. Because I remember whenever I learned in Gen Chem 1 what a mole was. Oh, 1 mole is 6.02 times 10 to the 23rd of something. That number was just entirely too abstract for me to understand. Um, and so it's really kind of tough for me to wrap my mind around that. Um, because then I was like, but what about a single water molecule? Is that a mole? No, it's not. Okay. It was tough for me to understand. And it kind of made me think about biology and chemistry at a single molecule level. And it seemed to me like, well, that's impossible. But the way that aquaporins work is they're really cool in the sense that it's these multiple polypeptides that fold together in a particular way that what they will do is look up here. I want to draw your attention to this space right here. Consider this to be the outer cavity or the basically near the outer leaflet. So the space outside of the cell. What these polypeptides will do is they will fold in a particular way to enable a single water molecule at a time through. Essentially, if you look at these water molecules right here, those water molecules, they are all positioned, and I always kind of think about it as like the door to a first grade classroom or maybe kindergarten classroom. The teacher always says like, okay, single file line. Or the teacher will say, let's get in a single file line. And every first grader rushes to the door. That's like all of the water molecules rushing to the door. They're rushing to that pore. And then what the teacher does is the teacher's like, okay, well, our line leader this week is Joe, and then it's Sally, and then it's Danny, and then it's Chris. Okay. So we have like the teacher effectively is putting everyone in that right position. And so when they are walking through the doorway, the teacher is checking, okay, do we have the sequence one person at a time? And is everyone facing forward? Is everyone in their proper position? And that's effectively what this protein does. And the way that it does it is with different amino acid interactions. So here, what we have are a couple of different amino acids that kind of reposition that water molecule. Now, what is kind of unique is that much of the pore is lined with hydrophobic amino acids. So what are hydrophobic amino acids going to do? They're going to repel water. Yeah, they're going to repel it, but another thing that they're going to do is they're kind of going to put a force against against that water molecule. Now, if you've got a hydrophobic force pushing this way, and then on the opposite side of that water molecule, maybe a oppositely charged ion interaction pushing in the other way, basically that water is kind of like forced in a specific direction. And when it's forced in that one designated direction, it doesn't really have a choice. And, if it turns to the left or turns to the right or anything like that, it has to go a single file. And so that's what's going to happen with an aquaporin. And so this to me was really kind of a cool and helpful thing to get to, oh, this is a single molecule of water that's moving through. When I've been thinking about moles and concentrations, this is a singular water molecule that moves in this one direction, and it has to go one molecule at a time. Now, with all that in mind, these aquaporins are a pretty cool area of research for people who are looking into like water purification systems, because this is a way that you can purify water because these aquaporins, they're not going to let a potassium ion, they're not going to let a sodium ion or chloride ion through. They're going to be very selective. They're going to say water only. So at the tightest point, each water molecule is separated from any other water molecules. So the primary reason for that is interactions with arginine and asparagine. So those are two, arginine's a polar positive and asparagine is a polar uncharged residue. Now, I highlight this, and this is just one of the important reasons that throughout the course of the semester, I've really tried to stress the importance of understanding and knowing your amino acid side chains and knowing your proteinogenic amino acids. Because, yeah, the better you understand this, then you can say, oh, okay, that's exactly how that residue is classified. So if we didn't have that, then those water molecules can move at a faster rate and with less organization. Now, ion channels are channels or proteins that move specific ions from one side of a membrane to the other. Ion channels are specific for particular ions like sodium, potassium, and chloride. And what they're going to do is kind of the same thing. They're going to position a ion. They're going to use water to solvate a potassium ion, and that potassium ion will be brought to basically its active site. And the potassium ion is going to select for potassium over any other ion. Now, these are going to move these ions at a very, very high rate. Now, GLUT transporters, these act in a little bit of a different fashion, and these are kind of simple to understand. Um, we've, in part because we talked about these GLUT transporters very early in the semester when we were talking about glycolysis. Your GLUT transporters are going to bind glucose and they're going to close off, and then they're going to release glucose on the other side of the membrane. So they'll take a glucose from outside of the cell, bind it, close off, which is what we see here, just closing off, and then they will release them. Now, these GLUT transporters are extremely selective. There's a couple of different GLUT transporters. They undergo a conformational change, hence why they close off. They're specific for glucose or for fructose. What they do not bind to at all is glucose 6-phosphate. They have very, very, very low affinity for glucose 6-phosphate, which is important because glucose 6-phosphate is a molecule that's effectively, well, if it needs to get out, it needs to be dephosphorylated. Otherwise, it's going to just go through the rest of glycolysis. Now, we've got active transport, and then we're going to tackle a sodium and potassium channel, and then we'll get to a little bit of metabolism. Okay, energy input drives transport. Some transport is going to flow against a thermodynamic potential, in which case energy input is going to drive that transport. So energy source and transport machinery are coupled. So here what we have is primary active transport, where one molecule, these X's, are moved against their they're moved against the concentration gradient. X is at a low concentration inside the cell to a high concentration outside the cell. Well, that's not going to spontaneously occur. So hydrolysum ATP moves it across. Done. Now, secondary active transport, we've got two players here. We've got X and we've got S. Both of them are moving against the concentration gradient. So how's that going to happen? Well, if we look at S first, S is going into the cell. S, I'm going to say in, is unfavorable. X going in is favorable. So here what we have is what's known as a symport. two molecules moving in the same direction. How does that happen though? Because X moving into the cell is good, it's easy. It's moving with its concentration gradient from high to low. S, by contrast, is moving from low to high. So it's almost like the favorability of X going in is able to like decoy and bring S in as well. Now, in order to keep this system going, what we have to do is maintain a low concentration of X inside the cell. How do we do that? X in, or low, I'll say low X in, bracket this, is done with this protein right here. Done with ATP hydrolysis. So that ATP hydrolysis helps X move from a low concentration to a high concentration. And that's a mechanism to bring S into the cell. So this secondary active transport is really, if you look at this second model, you say, well, what's the point of this? The point of this is to get S into the cell. How does S get into the cell? Well, it gets into the cell with the help of X. And so that SYM port, it's not hydrolyzing ATP, but this uniport over here is. And that hydrolysis of ATP is it's like it's trickling down in some respects to that SYM port to help that SYM port work. Now, sodium and potassium channels. These sodium and potassium channels, they are important for regulating kind of an electrical potential difference from the inside and the outside of the cell, your membrane potential, regulating concentrations of potassium inside and outside of the cell. That's an important part of basically your heart working and any other muscle contracting properly. So this type of protein maintains low intracellular sodium and high potassium levels. Okay. Now, this is crucial for all organs, but especially for neural tissue and the brain and the heart. ATP hydrolysis drives sodium out and potassium in. So how does this happen? So there are 3 positive charges exit for every 2 that enter. So 3 sodium out for every 2 potassium in. 3 sodium out, 2 potassium in. 3 sodium out, 2 potassium in. Now, all cells expend a huge amount of energy in order to do this. What does this look like from a mechanistic standpoint? I'm going to bring all this up. Here, what we have is a protein, or our protein. And I'm going to start up here. Whenever you see one of these kind of cyclical models, it's helpful to, think about, well, I have to start somewhere. Now, E1 here is, E1 is okay. I'm going to pause for a second. I'm going to make questions on this exam and on the final that have to do with lipid metabolism bonus. So that's what I'm going to do. So I'm going to ask you to, and I'm going to tell everyone in an announcement, lipid metabolism, just look at that on your own. Those are all going to be bonus questions. Okay, so now I'm going to spend a little bit more time on this. And I apologize for that. That's not what I wanted to do, but that's opportunity. Opportunity for some bonus points. Okay, so our enzyme, our sodium potassium ATPase, we'll look at that as E1. What that molecule is going to do, and this is on the inside of the cell, it is going to bind to a total of 3 sodium ions. These 3 sodium ions, whenever E1 is bound to that, will also bind to ATP. Okay, so E1, or our enzyme, whenever it has 3 sodium ions bound, it's going to bind ATP. And whenever it binds that ATP, it's going to hydrolyze that ATP and transfer a phosphate group onto itself and make an aspartyl phosphate. So it's going to transfer that phosphate onto an aspartic acid. Now, transferring that phosphate onto that aspartic acid is this unique form of our protein where it's going to undergo a conformational change. When it undergoes that conformational change, then it can release its three sodium ions on the outside of the cell. You could kind of think about this as a ping pong mechanism. So we bind our sodium ions and ATP. We make our different form of our enzyme, then they release one of our products. Okay, those three sodium ions. When those three sodium ions have been released, you have our phosphorylated enzyme that's not bound to any ions. What it's going to do is it's going to take on two potassium ions. Those two potassium ions, whenever it binds those, it can release our inorganic phosphate, our PI. And then we have our dephosphorylated protein with two sodium or two potassium ions bound. It's going to undergo another conformational change and release two potassiums on the inside of the cell. So essentially, protein binds ATP, binds sodium, binds ATP, phosphorylates itself. There's my phosphate group. So it's got sodium and a phosphate group. Then it undergoes a change where it can release the sodium and then it can bind potassium. Still phosphorylated, but now that it's bound to potassium, it can dephosphorylate itself. So this is one that I think is helpful to think about kind of a sequence. One thing that's always confusing is this E1 and E2. Don't get lost on that and think like, oh, well, this is a different protein. It's the same protein, but it's kind of like a different form. Think back to the T and the R state of hemoglobin. It's the same thing, but it's got a different shape. So bind Na plus, bind ATP, phosphorylate self. 4, shape change. Actually, I'm going to add to this phosphorylate self, release ADP. Shape change. Okay, so then our shape change, release Na plus out. Then 6 will be bind K plus. 7 will be dephos self. And 8, release K plus in. So that's effectively the sequence of events that happens with the sodium and potassium channel. If you're thinking, what do I need to know about this? I would start with, what am I doing? What's the purpose of this? The purpose of this is to get sodium outside of the cell and to get potassium in. So if you think about this in terms of, and I always think about this in terms of endurance events. From time to time, and this is always sad and catastrophic, but from time to time, if somebody's running a marathon or something like that, what they might do is they might have a heart attack. And I think that it's common for people to think like, oh my gosh, it's because of the endurance event. It kind of is, but that heart attack in some instances is caused by basically a mineral imbalance. A person, maybe what they were doing at every one of their aid stations, they were probably sweating a lot. So they're getting, their body's losing lots of electrolytes. They're getting rid of lots of sodium. And then what they're doing is maybe they're just drinking water. And that water, unlike a Gatorade or, other electrolyte mixture, other electrolyte drink, doesn't have potassium. So you're losing sodium, but you're not replacing it with an adequate amount of potassium. So you need to be continuously bringing in that potassium so that channels like this can work. And in some instances, that causes muscles to seize up, and unfortunately, in many cases, that can cause a heart attack. So There we go. So know that sequence of events, like what's going to happen where. Is the phosphorylated form of the protein going to bind to the potassium, or is it going to bind to the sodium? Dephosphorylated form, what's that going to be bind to? And if it's on the inside or the outside, what's it going to do? Okay. So that ultimately plays into secondary active transport for moving glucose. Here, what we have is a bloodstream in the intestinal lumen. So in your gut, you're eating a bunch of food, you eat a steak and you put some honey on that steak. You get glucose from that honey and sodium ions from the steak itself. Those sodium ions are going to go into an epithelial cell with the help of a sodium glucose symporter. So the sodium and glucose both make their way into the cell. A GLUT transporter, a GLUT uniporter, is going to take that glucose and put it into your bloodstream. Now, within your bloodstream, you're going to be bringing in 3 sodium ions, and you're going to be getting those potassium ions to go on the opposite side. So how you have a couple of different things going on here with how you get basically the sodium and glucose from your intestinal lumen into your bloodstream. And that's where I'm going to stop. Now, or that's where I'm going to stop with like kind of a lecture. In terms of lipid metabolism, all these questions on your exams are going to be bonus points. So I want you to think about that and be aware of that. I'm going to send out an announcement. Now, what questions should you think about? Or what should you be approaching? Products. Products of beta oxidation. What are they? Acetyl-CoA and a shorter fatty acid-CoA. In addition to that, FADH2 and ADH. Then, if you were metabolizing a fatty acid that's 16 carbons long, how many rounds are you going to go through? You're going to go through 7 rounds. How many ATP are you going to get from that? Like 130. So you're only going to go through, if you have an 18 carbon long fatty acid, you're going to go through 8 rounds. So spend a little bit of time on this. But like I said, the questions that pertain to beta oxidation are all going to be bonus questions on both this 4th exam and your final exam. I'll see what videos I have from previous semesters that explain beta oxidation. So I will post those if I find something. Other than that, I hope you guys have a wonderful day. And thank you very much for coming to class. And if anything comes up between now and Wednesday or between now and next Monday, please don't hesitate to contact me. All right, everyone. Thanks for coming. See you guys.